So the types of the gene therapy, there are two: the germline gene therapy and the somatic gene therapy. So when we say germline gene therapy, it is the permanent changes. And when we say somatic gene therapy, it was affecting only the targeting cells in the patient, is not passed to future generation. So in germline gene therapy, it has a potential for offering a permanent therapeutic effect for all who inherit the target genes. It also the possibility of eliminating some disease from a particular family. And the types of the somatic gene therapy, which is ex vivo and in vivo. So when we say ex vivo, it is modified outside the body and then transported back it again. And when we say in vivo, gene are changes in cells when the cells are still in the body. Why it is called ex vivo or in vivo? So when we say ex vivo, it was treated outside of the body. And when we say in vivo, it was transferred to the cell inside of the patient body. So there are the basic process of gene therapy. There are two categories, which is viral vectors and non-viral vectors, which is the viral vectors is recombinant viruses, and the non-viral vectors is the naked DNA or DNA complexes. So these are the example of ex vivo and in vivo. As we can see here in the side of this, there is a therapeutic gene inserted or injected to the patient. And we can see here the in vivo, it was a therapeutic gene packaged into delivery vehicles such as the retroviruses introduced to the cells, then transferred to the patient. Example of ex vivo gene therapy. First is the gene therapy to correct deficiency of enzyme, which is adenosine deaminase or ADA. Performed on the 4-year-old girl. For example, this is the true story of Ashanti De Silva. She was suffering from SCID, which is severe combined immunodeficiency. It's caused due to defect in gene coding for ADA, which is adenosine deaminase or ADA. Deoxygenases accumulate and destroy T lymphocytes, disrupt immunity suffering from infectious disease, and die at a young age. So will we tackled about in vivo gene therapy? So it can be the only possible option in patients where individual cells cannot be cultured in vitro in sufficient numbers. Example of that is brain cells. In vivo gene transfer is necessary when cultured cells cannot be reimplanted in patient effectively. Example of in vivo gene therapy, um, in the patient with cystic fibrosis, a protein called cystic fibrosis transmembrane regulator or CFTR is absent due to a gene defect. In the absence of CFTR, chloride ions concentrate within the cells and it draws water from surrounding. This leads to the accumulation of sticky mucus in respiratory tract and lungs. Treated by in vivo replacement to defective gene by adenovirus vectors. So let's proceed to the therapeutic gene. So we have monogenic diseases. So when we say monogenic diseases, it is the result from modification in a single gene occurring in all cells of the body. So we have a healthy gene will be introduced, which is hemophilia, cystic fibrosis, and multifactorial diseases, which is also categorized as a diabetes, cancer, AIDS. So there are two categories of this. Interaction in more than one gene, which is we have gene A gene B, and gene C. And into the environmental factors also, we have the infections or diet or lack of the exercises. Next is we have vector in gene therapy. To transfer the desired gene into the target cells, a carrier is required, such as vehicles of gene deliver are known as vectors. So there are two main classes of the vectors, the viral vectors and non-viral vectors. So ideal vectors, it is targeted the right cells, integrate the gene in the cells, activate the gene, avoid harmful side effects. No universal vectors exist. So the example of that are here, as you can see. So let's proceed to the two classes of vectors, which is viral. Viral vectors, it is a virus is introduced their genetic material into the host cell as part of their replication cycles. Remove the viral DNA in using the virus as a vehicle to deliver the therapeutic DNA. The viruses used are altered to make them safe, although some risk still exist with gene therapy. So the example of viral vectors are like this. So this is the viral vectors carrying new gene. So as we can see, here the viral vectors inserting then you can see the gene inserting inside, then you can see here it's fulfilled. So there are also types of viral vectors. A number of viruses have been used for human gene therapy, including retroviruses, adenoviruses, adeno-associated viruses, herpes simplex virus. So when we say retroviruses vector system, it is the recombinant retroviruses have the ability to integrate into the host genome, can carry a DNA of size less than 3.4 kb and target cells dividing. When we say adenovirus vector system, it is the adenoviral DNA does not integrate into the genome and it is not replicated during cell division. Humans commonly come in contact with the adenovirus. Majority of patients have already developed neutralizing antibodies which can inactive the virus. So the target of adenovirus vectors is the non-dividing and dividing cells. Third is adeno-associated virus vectors. It is a human virus. It is a single-stranded AEV enters host cells, becomes double-stranded, and gets integrated into chromosomes. AAV, which is adeno acid associated virus, is not currently known to cause disease, and consequently the virus causes a very mild immune response. So the target of this is non-dividing and dividing cells. Fourth is herpes simplex virus vectors. Viruses which have natural tendency to infect a particular type of cells. The herpes simplex virus is a human neurotropic virus. This is most examined for gene transfer in the nervous system. So there are also the advantages and disadvantages of this. So first, the advantages: targeted target a specific type of cells first, and they are very good at targeting and entering cells. They can be modified so that they can replicate and destroy cells. The disadvantage of this is they can cause immune response in the patients. They can carry a limited amount of genetic material. Therefore, some genes may be too big to fit into some viruses. So next is we have non-viral vector system. Pure DNA construct. So Meaning of that is direct introductions rather of pure DNA construct into target tissues. Efficiency of DNA uptake by cells and expressions rather low. Consequently, large quantities of DNA have to be injected periodically. And the second one is DNA, molecular uh, con- conjugates rather. It is commonly used synthetic conjugate is polyl lysine bound to specific target cells receptor. Therapeutic DNA is then made to combine with the conjugate to form a complex. It avoids lysosomal breakdown of DNA. Third is lipoplexes. When we say lipoplexes, it is a lipid DNA complexes. It is DNA construct surrounded by artificial lipid layer. Most of it gets degraded by lysosomes. The fourth one is the human artificial chromosomes. It is a it is or it can carry a large DNA. Example of that is with one or more therapeutic genes.